Welcome to this introductory video on the recombinant DNA techniques. In this video, we will explore the core concept of rDNA and the recombinant DNA techniques such as polymerase chain reaction, RT-PCR, and DNA sequencing. The underlying principles and application of these techniques will be discussed briefly. However, for a detailed insight into the method, please check the dedicated videos available on our YouTube channel, Future Academics. To understand recombinant DNA techniques, it is essential to grasp the concept of rDNA or recombinant DNA. rDNA is normally created by cutting and pasting two unrelated pieces of DNA. One is vector, other is exogenous gene or DNA. Thus, rDNA can be defined as a DNA which is made up of two unrelated pieces of DNA. One is vector, other is exogenous gene. This strategy is commonly used in gene cloning and rDNA technology. The techniques that are used to construct and characterize the rDNA are known as recombinant DNA techniques or rDNA techniques. So rDNA techniques, there are three techniques. These are the techniques which are used to construct and characterize recombinant DNA. These are polymerase chain reaction or PCR. Second one is RT-PCR, in which an additional step of reverse transcription is introduced before PCR amplification. This is commonly used for amplification of RNA or RNA viruses. And the third one is Sanger's DNA sequencing method. The applications of these techniques are enormous. However, I have restricted myself to their application in clinical sciences. The first one is they are heavily used in understanding the molecular basis of disease pathogenesis. They are also used heavily in developing diagnostic tests or tools. You have seen recently use of RT-PCR in the diagnosis of COVID-19. And third one is in developing molecular treatment strategies, including DNA, RNA vaccines, and gene therapies. In summary, these techniques therefore are used in understanding one, molecular basis of diseases, two, in developing diagnostic tools, and third, in developing molecular treatment strategies. The general underlying basic principle of these techniques, they have been developed based on the basic principles of DNA replication or DNA synthesis and nucleic acid hybridization. Nucleic acid hybridization refers here to the binding of a primer and the target DNA or the template DNA used in these techniques. RT-PCR, whereas, includes an additional step or reverse transcription before its coupling to the PCR amplification. So thank you very much for watching this introductory video on the recombinant DNA techniques. I urge you, I request you to like it, share it, and subscribe it. For a detailed insight, I advise you to watch out our dedicated videos on our YouTube channel called Future Academics. Until next time, you take care, bye bye, and thank you once again.